Okay, folks, we're going to move on to that other part of the adaptive side of our immunity, and that is the cell-mediated immunity. Here we're not talking so much about antibodies. Here we're talking about those other lymphocytes. All right, B cells made the antibodies. They were part of humoral immunity, still specific, but humoral. And now we're talking about the T cells. We're turning our attention to those T cells that circulate through blood and the lymph. And these T cells, like the B cells, become immunocompetent. In other words, they too go to college and they become competent to be part of the immune system in the thymus. All right? So T for thymus, T for T cell. These cells, in comparison to B cells, need an antigen-presenting cell to actually be activated. Okay? B cells can run into antigens in the fluids, right, and start churning out antibodies, but T cells, they need to be presented with antigens by other cells of our body. These antigen-presenting cells could be macrophages or other phagocytes. Keep in mind, phagocytes gobble up these bacteria and they have some dirt on these pathogens, right? Because after they've digested a pathogen, they're going to have little pieces, little particles, proteins that have specific shapes that they could present to the T cells. Okay, and that's exactly what they do. These macrophages can do that. B cells can even do that. And these foreign fragments, when presented to a T cell, will really raise red flags, and these T cells will become much more active. Okay? Now, one kind of T cell that can be activated by an antigen-presenting cell is a cytotoxic T cell. Now, they receive activation from body cells. Okay? So let's say this purple cell here is a kidney cell. We'll put a K for kidney cell here. Or it's a liver cell, or it's a skin cell. Doesn't really matter. This would be the cell that is presenting antigens on its surface. And then this other cell down here is, let's say, a cytotoxic T cell. It's receiving signals because of the presentation of proteins on the surface of that kidney cell. Now, what cytotoxic T cells do is they identify and kill the abnormal and infected kidney cells or other body cells. They are not cells that are specializing in bacteria or virus, or I should say bacteria or some other kind of eukaryotic worm of some sort, right? These are specifically finding infected or abnormal body cells. Okay? Now, ultimately, if a cell is really bad off, right, it's abnormal or infected, then we might better get rid of that cell. And that's exactly what these cytotoxic T cells are capable of. They poke holes in the membranes. They would digest the DNA inside that body cell, trigger a mechanism within the cell for it to kill itself, called apoptosis, and they'd even call in the garbage men to pick up the pieces at the end. Okay, the macrophages will be activated. Now we're going to talk more about these cytotoxic T cells in a second, but I just wanted to show you a picture of what it can look like. This is a cancerous cell here. Cancer cells are abnormal for sure, right? And this is a cytotoxic T cell identifying something abnormal about this antigen-presenting cell and then killing that cell here. And you notice this cancer cell ends up in little pieces, little pockets of membrane. So it's not like the contents of a cell that's being destroyed are going to spill out. They're not going to affect other cells around it. So it's really kind of an interesting process. All right, so what are some of the other kinds of T cells out there? Well, there are helper T cells, which I put in bold here because they really don't get enough credit by just thinking of them as helpers, right? Well, somebody helped me do something. That suggests that I did it and somebody else kind of chipped in, right? Well, these helper T cells are really the hub of the immune system. They greatly increase immune immunity efficiency, and we will address them in a second. They're very important to activating and stimulating the entire immune system. We have just the opposite. We have regulatory T cells, which will limit immune activity. And if you think about it, it's almost just as important that we have this balancing act, right? Because when I don't have an infection, I want the energy that I have to be put towards other things, right? When I am infected with something and I'm sick, well, I'm drained, right? I feel drained because a lot of that energy has been put into the immune system. So we need the ability to toggle back and forth to use energy in different ways. And the nice thing is we do have memory cells that we can get from T cell stimulation, right? So let's take another look at this body cell to T cell presentation. The one we just got done talking about, right? So we have a body cell presenting to a cytotoxic T cell. And here there are some details I'm going to need you to know, all right? So here we have the cytotoxic T cell. Here we have the body cell, and they're talking about it as the target cell. Must be there's something wrong with the cell, right? It's abnormal or infected, so they're calling the body cell a target now. So here we have a cytotoxic T cell. Here we have the target cell. And they're coming into contact with one another, and there's various items that are involved here, okay? There are various proteins that we need to know about that are involved with this interaction, all right? There are a couple of proteins on the cytotoxic T cell side, and there are a couple of proteins on the target cell side, okay? So here I have a drawing that I put together. Hopefully it helps you, okay? So here we have a body cell. Let's say that body cell is infected. So here we have a purple people eater, right? This is a virus, let's say. And it has to get into a cell to survive and replicate, right? So there it is inside this body cell. It's infected that cell. And let's say that body cell is going to act as an antigen presentation cell. What it's going to do is it's going to use something called an MHC. This is a self-antigen for the body cell, but it's using an MHC class 1 protein here in red as a pedestal for this purple antigen that's part of the virus. It is presenting it in that way. Okay. Now on the T cell side of this whole ordeal, there's a T cell receptor. Okay, and it is receiving the signal of that viral protein. This cytotoxic T cell is also going to reach across and just check out this MHC. Almost like here we have a spy, right? This body cell is a spy for my body, and it is bringing back this piece of information to show it to the immune system. I want to be able to check out that body cell and be sure that it is in fact one of my own spies, right? So there's a couple things we're checking out. We're checking out the protein being presented, and we're checking out the MHC being used as a pedestal. There are 3 ways this cytotoxic T cell may get all hot and bothered and kill that body cell. One is the presentation is a presentation of a cancerous antigen. A cancerous protein sits there on that pedestal. That would be cause to kill that body cell. A second thing, if that is indeed a viral protein, that would be cause to kill the cell, like in our scenario we have here. And there's a third possibility, and that is if this MHC complex is not a familiar complex to the cytotoxic T cell. In other words, if this, let's say this is a kidney cell here, right? So this is actually a kidney cell. If this kidney was a transplanted kidney from someone else, that would be cause for the cytotoxic T cell to reject that cell. And this is exactly the reason that we need to be concerned with these things when we get an organ transplant, right? So 3 ways a cytotoxic T cell will recognize that this cell is infected. All right, what about helper T cells? So here we're going to focus on the helper T cells, where an immune cell will present to the T cell. Okay, so in this case, we're talking about helper T cells, and not just any old body cell, but an immune cell, and like a white blood cell is going to do the presentation. So here we have our antigen presenting cell, and let's say this is a macrophage. Okay, it's gobbled up bacterium, turned around, it's going to present a piece of that bacterium. And here we have our helper T cell. So we have a similar situation. We have a T cell receiving information from an antigen presenting cell. In this case, it's an immune cell, a macrophage. Okay. So let's look at my kind of silly drawing here, but hopefully it helps. Here we have our phagocyte. It just went through phagocytosis, gobbled up something. And over here on the other side, it's working as an antigen presenting cell using, again, an MHC complex. Pretty similar, right? Except this is a MHC2 presenter. Now on the surface of that, as like a pedestal, it's holding out this bacterial antigen, let's say, right? That's on the surface. And on the other side, we have our helper T cell. Again, a T cell receptor is being used, and the antigen is being checked out, right? This helper T cell has gone to college. It knows that that's a foreign antigen. Now, like the cytotoxic cell did, there is another protein, in this case a CD4 protein, that is reaching around to check out the MHC protein. Now, if that particular antigen is foreign, like it is in our scenario here, then that helper T cell is going to activate all sorts of cells in the immune system. It's going to activate phagocytes in the region, macrophages, neutrophils. It's going to make memory T cells. Great, I want that, right? I want memory so that I can have this building of an immune system, just like we had with B cells. And it's going to activate B cells that have come into contact with the very same antigen. After all, that B cell would not have been selected to be cloned unless it made contact with an antigen that fit its antibody. And it too will make plasma cells, memory B cells, and we're better off for it. So what we've learned here is that not only do CD8 cells, of course that has to be the cytotoxic ones, right, because they're the only ones with CD8, and CD4 cells has to be helper T cells, right? Not only do they divide and give us more activity, but they also give us memory cells, right? Which is exactly what I want to have a robust immune system, one that works for me in the future. But it's those helper T cells that are really central to this whole deal. The helper T cells can communicate. They use cytokines, or interleukins, they will call them sometimes. Interleukins, I always like that better. That word interleukin is between white blood cells, right? Inter for between, leukin for white blood cells. And they can use this to talk to non-specific side of the immune system, the humoral side when it comes to antibody production, and the cellular side. So this helper T cell, even though we think of it as the part of the cell-mediated part of the immune system, it really is going to activate across the board. So it's central to the acquired immune system. When we lose the helper T cell, which does happen with HIV viral infection, we know what happens, right? The immune system really falls down, really fails. So here I have, with, again, kind of my silly drawings here, the whole ordeal, right? We have all of this going on with a specific immune system. We have these different chemical mediators that are going to allow the cells to talk to one another. And in this case, when we're activating the immune system, they all have a plus sign next to them. These cytokines or interleukins are all activating and stimulating this immune system of ours. Okay. If there were regulatory T cells in this picture, you would see that there would be inhibitory cytokines, where it would kind of cool everybody down, kind of, okay, we don't have an infection, let's relax, right? Now, one last thing I'll mention before we call it quits on this particular clip. We've talked about cytotoxic T cells and helper T cells. And we noticed that the cytotoxic T cells will recognize something about something that's inside of the cell, right? Like a virus, for example. So when we talk about cell-mediated immunity, oftentimes we bring our attention to that cytotoxic T cell. It's recognizing something inside of our own cells, okay? It's recognizing something inside, like a virus. So the cytotoxic T cells are really about the intracellular pathogens more than extracellular pathogens, those cytotoxic T cells. When we talk about extracellular pathogens, well, we focus a little more on the humoral side, okay? We refer to B cells and the making of antibodies and all of that business, right? In the center of all this is where we're going to have our helper T cells, okay? Because helper T cells are going to receive signals and give signals to the humoral side, and they're going to receive signals and give signals to cytotoxic T cells as well, okay? So here we have at the center our helper T cells, our cytotoxic T cells worried about intracellular pathogens like viruses, and our humoral side with our B cells and our antibodies concerning themselves more with extracellular pathogens. Okay? All right, so we're going to call it quits, and we're going to look a little bit at immunodeficiencies and immune system disorders and things like that next.